Oh, hi, thank you for the introduction, so I didn't have to read all of that. So yeah, we'll be talking about COVID-19 and pregnancy today. And I have no conflicts of interest to declare, so yay. Starting with COVID-19 and pregnancy, pregnant women with COVID-19 are at higher risk of adverse clinical and pregnancy outcomes. And early in the pandemic, there were no PK data for COVID-19 antivirals in pregnancy. So to this end, IMPACT 2032 was performed to evaluate the safety and PK of Remdesivir in pregnant and non-pregnant women with COVID-19. So Remdesivir is a nucleotide prodrug. It's administered intravenously because it undergoes extensive first-pass metabolism. After administration, Remdesivir loads cells and undergoes a series of hydrolysis and phosphorylation steps to yield its active triphosphate form GS-443902, which I'll just be referring to as 902 throughout the presentation. So 902 competes with ATP to inhibit SARS-CoV-2 RNA-dependent RNA polymerase. 902 concentrations in PBMCs are often used as a surrogate measure for Remdesivir activation. However, prior to IMPACT 2032, DBS concentrations of 902 had not been reported. In the primary analysis, the focus was primarily on Remdesivir, the alanine metabolite, and the nucleotide metabolite, or the nucleoside analog, in plasma. So this follow-up analysis is looking at 902 concentrations in DBS and PBMCs. So looking at the previous results, the plasma PK of Remdesivir and its major metabolites were similar between pregnant and non-pregnant women. The clinical significance of this is not very clear at this time. They also found accumulation of 902 in PBMCs over time differed by pregnancy status. So as you can see in the figure in blue, the non-pregnant participants tended to have an increase in 902 concentrations in PBMCs as the time post-Remdesivir initiation increased, whereas the pregnant participants tended to have a decrease or almost a plateau in those 902 concentrations. So with this brings us to our objectives. Since the primary analysis found differences between pregnant and non-pregnant PBMC concentrations, we wanted to further investigate factors associated with the intracellular concentrations of 902 in PBMCs, and then also look at DBS. And then we wanted to look at the relationship between DBS and PBMC concentrations to see if DBS may provide an alternative matrix to evaluate Remdesivir activation, since it's an easier matrix to work with and a little more cost-effective. So I guess going back through, IMPACT 2032 was a phase 4 prospective open-labeled opportunistic study of Remdesivir in hospitalized pregnant and non-pregnant women with COVID-19. So Arm 1 consisted of 26 pregnant women. Arm 2 consisted of 28 non-pregnant women. Baseline data were collected 48 hours prior to Remdesivir initiation, and then during the infusion period, participants received IV Remdesivir for up to 5 to 10 days. Sparse PBMC and DBS were collected pre-dose and then 23 hours after the end of infusion on day 3, 4, or 5 for each participant. So PBMCs were previously analyzed by QSP, and cell counts were based on DNA quantification. 902 concentrations were directly quantified using a validated LC-MS method. DBS were analyzed at the Colorado Antiviral Pharmacology Lab. 2 7mm punches were used for indirect quantification of 902 concentrations, again using a validated LC-MS method. So participants with at least one intracellular concentration were used for the PK analysis population in this, which resulted in a total of 25 pregnant participants with 37 unique observations and 26 non-pregnant participants with 39 unique observations. To investigate the factors associated with the intracellular concentrations, linear mixed effects modeling was used to account for the repeated measures. The pre-infusion baseline data were screened to include physiologically relevant covariates with less than 10% missing, and then following this, a covariate search was performed with a forward selection, backwards elimination method to identify the covariates significantly associated with the intracellular concentrations. And then looking at the relationship between DBS and PBMC concentrations, linear mixed effects modeling, again, was used with stratification by pregnancy status. So looking at the baseline demographics, the non-pregnant participants tended to be slightly older and weigh more compared to the pregnant participants, and they comprised mostly of Black women. The majority of women were on low-flow or high-flow oxygen therapy at baseline. And then consistent with physiologic changes in pregnancy, EGFR and ALK Phos were elevated in the pregnant participants compared to the non-pregnant participants. Systolic blood pressure tended to be lower in the pregnant participants compared to the non-pregnant participants. And white blood cell count and platelet count were both elevated in the pregnant participants compared to non-pregnant participants. So starting with the DBS results, looking across the Remdesivir infusion period, it appeared that the DBS concentrations of 902 in the pregnant women tended to be higher compared to non-pregnant participants. If you look at the pooled concentrations, they appear to be similar. However, when you account for infusion number, systolic blood pressure, ALK Phos, and platelet count, there was a roughly 20% decrease in 902 concentrations in the DBS for the pregnant participants compared to non-pregnant participants. Additionally, for every 10-unit increase in systolic blood pressure, there was a roughly 8% decrease in 902 concentrations in DBS. A 10-unit increase in ALK Phos was associated with a 1% increase in DBS concentrations, and a 50-unit increase in platelet count was associated with a roughly 5% increase in DBS concentrations. So then looking at the PBMC concentrations, as I described previously, there's a concentrations in pregnant participants tended to be lower compared to non-pregnant participants as time after remdesivir initiation increased. And then we found a significant interaction with pregnancy status and infusion number. So as with each additional remdesivir infusion, there's approximately 45% reduction in PBMC concentrations for pregnant participants compared to non-pregnant participants. Additionally, a 10-unit increase in ALK Phos was associated with a roughly 4% decrease in PBMC concentrations, and a 10-unit increase in ALT was associated with an 11% decrease in PBMC concentrations. So, like I said previously, we want to probe the relationship between DBS and PBMC concentrations. However, before accounting for pregnancy status, no relationship between these measures was observed. So then when you account for pregnancy status, you see kind of opposing trends between pregnant and non-pregnant. So for every 100-unit increase in DBS concentrations, there's a roughly 8% increase in PBMC concentrations for the non-pregnant participants, whereas for the pregnant participants, for every 100-unit increase in DBS concentrations, there's roughly 4.4% decrease in PBMC concentrations. And the differences in the PBMC loading is likely what's driving the differences we're observing here. So with this, the cellular disposition of 902 differed by cell type and by pregnancy status. The intercellular concentrations of 902 were influenced by measures associated with COVID-19 disease severity and pregnancy status. However, the mechanisms and kind of clinical significance of this kind of needs further investigation at this point. So with this, I'd like to make the acknowledgments and thank the Impact Study team and Gilead who sponsored this study and all the participants of the study. And thank you.